Okay. RTB, Reason to Believe, is an international ministry founded by Hugh Ross, a Christian astrophysicist, in 1986. Reason to Believe uses scientific advances to answer questions and identify new evidence of God's existence, character, and the Bible's reliability. Reason to Believe engages statistics and engages skeptics and cultivate communities where scholars and others learn to integrate science, theology, and philosophy so they can open the world of science to the gospel. And today we want to thank Dr. Mancha to come to talk to us on part 1 of the epigenetic, which is just a review of biology, cell 101. Dr. Mancha has an MD from University of Alabama at Birmingham with an undergraduate degree in biology from Florida State University. And he's a retired board-certified family practitioner and presently resides as a director of Mobile, Alabama, Rachel Christie, and the president of Darwinian Alabama chapter of Reasons to Believe. His apologetic interest is primarily in evolution and is eclipsed due to non-Darwinian epigenetics. So take it over, Dr. Mancha.
Well, okay, well, thank y'all very much. And in addition to the other things, I also did 15 years of clinical research. It's one of the articles on the bottom right that I was involved with publishing. I did about 40 of those. Excuse me 1 second here. The title of the talk is The Central Dogma, or Are We in a DNA Matrix? And on the left-hand side, you can see the slide, which basically shows the famous movie, The Matrix. And then you see a DNA double helix spinning around. In other words, are we just a part of the letters that come out of our DNA? Are we completely determined by our DNA, or is there something else involved? This is from the movie The Blade Runner. As you recall, Harrison Ford was trying to delete basically these guys called skin jobs. They were superhuman clones who had gotten off the reservation. And this one clone made it back to meet his boss called Terrell in his tower. And as he was talking to him, he said, "I've done questionable things." He said, "but nothing the God of biomechanics wouldn't let you into heaven for." And I thought that was a very philosophical statement. He went on to kill his own maker right after this, which is also very philosophical if you think about it. If you look at the book of Job, and this has been a 40-year project for me, I've discovered that the book of Job is not so much about undeserved suffering. It's really a debate between God and Satan over free will and whether the angels were just made the way they were apart from what God wanted. In other words, they really didn't have anything to do with things. If you've seen the first book, the first chapter, God said to Satan, "Have you considered my servant Job? He is blameless and upright." And then Satan quips back, does Job fear God for nothing? Have you not put a hedge, determinism, around him? In other words, God made him do what he's doing by hedging him into it. Job went through 2 tremendous trials, and both times came out wonderful. So he had 3 friends which stayed with him. For 7 days, they said nothing. And then one friend, Eliphaz, who's the eldest, had a vision during the night. And the vision said to him, "Can mankind be just before God?" This is Satan, was that vision, or ghost. "Can man be pure before his maker?" He goes on to say he puts no trust even in his servants, the fallen angels. He puts no trust in them, he accuses God. And against his angels, he charges error, basically determinism. How much more those who dwell in houses of clay, whose foundation is in the dust? So he ties us at the hips together that the angels were basically charged because he didn't trust them. And Satan is basically putting his appeal to God out in Job. It's a phenomenal thing to see that this is Satan's appeal, that God, you made me, therefore you're to blame. Later on in the book, Bildad, who's one of the other friends, said, how then can mortals be righteous before God? Again, quoting the hidden spirit. And they were assuming this was God. He said, even if the moon is not bright and the stars are not pure in his sight, is basically saying that they're entropic, and they're not really pure or perfect. He said, how much more a mortal who's but a maggot? So basically, he's equating the entropic state of the universe and the fact that it's wearing out to our sinful nature. So by assigning the necessary entropy, and I say necessary because for us to work, we have to have entropy. Food generates energy through entropy. And so it's necessary that our universe have entropy for us to live and move. But by assigning the necessary entropy of the universe to the entropic mutations in nature of our DNA under natural selection, do we have free will, or are we under determinism? The sum of our DNA evolved over billions of years. So this is the central dogma. It was described by Francis Crick. Francis Crick was the co-discoverer of the DNA molecule back in 1953. He came up with the central dogma theory in the late 50s, and this is a rehash of the article in 1970. And the picture on the right I took out of a magazine that did an article on him and said that he was a big-time LSD tripper, and they really think that he was probably on LSD when he discovered the DNA molecule. So that was just kind of a factoid. Nevertheless, on the bottom, the central dogma of biology, and I'll explain later, means that information goes from DNA to RNA to protein and not the other way around. And it doesn't go from protein to protein or protein back to RNA or DNA. And this is what I learned in biochemistry when I was in college, and I believed it, basically. I had nothing else to go on. This is kind of a summary of the central dogma on the left-hand side. You see that the DNA molecule forms an RNA molecule, which then forms a protein. DNA during replication can sometimes have mutations. And according to neo-Darwinism, they felt these mutations were random, and thus it could make random changes to the DNA, which would make random changes to the protein. And if the protein was better, natural selection would kick in. As it turns out, in just the last 5 years, we have discovered that all these mutation mechanisms of correcting these mutations are controlled by epigenetics, and it's biased mutations. It's not random. So for 75 years, experiments proved evolution by natural selection had to assume the central dogma as the heart of it. This is also another diagram of the central dogma where you have the cell nucleus in blue, and it has the DNA, which then transcribes into an RNA molecule, which then goes out into the cytoplasm of the cell. And that RNA molecule is then turned into a protein by ribosomes in green. Each 3 letters of the DNA codes for one codon, as it's called, and each codon codes for one amino acid. This idea was actually also proposed by Francis Crick, along with some other colleagues. And this is what makes up the genetic code. And it was basically discovered in the early 1960s. Here's an example of it. You can see 3 letters for each codon in the middle, where you got AUG is one codon, GUC is another codon, and these different codons make different amino acids. This is an example of the transfer RNA, which takes the RNA and takes one amino acid at a time and puts them together in a protein chain. You can see the codon UCA on the bottom, and it has one particular amino acid with that codon, and then it just manufactures these right along. As the amino acids go out in a chain, they fold up into a protein, and it has a very distinct shape and structure. When I was in school, this structure was supposed to be very precise, but we've later learned that this is not the case. So the protein comes out as an amino acid chain, and then it starts to take different shapes, like a helix, which is a secondary, and then there's a tertiary structure, and then a quaternary structure. On the bottom right, you can see the proteins put together, and you see little orange like Tic Tacs, those are the active centers of the protein. They are the things that do the work that causes change. So codons are made up of threes, and yet there's 4 letters in the DNA, A, T, C, and G. So if you take that 4 and raise it to the power of 3, that gives you a possible combination of 64 codons. But interestingly enough, there's only 21 amino acids for those 64 codons. Why is that the case? We really don't know. It's redundant. In other words, many different codons can make the same amino acid. So that, as it says, redundancy in the genetic code means that most amino acids are specified by more than one mRNA coding. This is a table you don't really have to memorize or anything, but it shows you groupings of the codons in threes. If you take the pink box on the top left, you see UUU, UUC, UUA, UUG, and then the next box down, all those 4 codons make the same amino acid, leucine. So in this case, leucine is coded for by 6 different codons. And so that was a mystery to people for quite a while. Crick had no explanation for why there would be 64 possible codons and only 21 amino acids. He just called it a frozen accident at the beginning of life, which is very non-curious, isn't it? He insisted, instead, he tried to use this to explain the genetics of neo-Darwinism in a post hoc fashion, and I'll explain how this happened. Per neo-Darwinism, redundant codons, they felt like, would not change the amino acid, and that's true. Therefore, they thought that the protein would be neutrally selected. You know, in Darwinism, he only talked about natural selection as though it was a positive thing. But later, they started saying, well, there's negative selection, there's neutral selection, there's positive selection. All kinds of selection, no matter what it is, it's selection. So they give credit basically, just, despite the form that it took. However, if a mutation changed the amino acid sequence, and that would change the protein, and then that would change the body, that change in the body would be under positive natural selection, or survival of the fittest. Excuse me. This is a concept that's a part of neo-Darwinism, and it's the concept of synonymous mutations and synonymous, non-synonymous. Synonymous means the same. So if you look at the leucine in purple on the top left, it has a UUA, and then a mutation went to UUG from A to G, and it still is a leucine because of the redundancy of the codons. But a non-synonymous mutation, below that, we have the leucine, a UUA goes to UUC, that causes phenylalanine, and it's a different amino acid, so it's non-synonymous. Neo-Darwinism for the last 60 years basically counted the synonymous versus the non-synonymous mutations, and they did ratios based on those to determine how much natural selection was going on at the genetic level. And I'm going to present an article later on showing how the things have changed so much that 60 years of neo-Darwinism is out the window. So here we have our DNA without mutation on the top, and you can have an insertion of a nucleotide or a deletion of a nucleotide, and that causes a different amino acid. And then you run it through the central dogma machine, the DNA going to RNA, going to protein, and that makes you a different animal, basically. Here we have a penguin who can jump further because of a mutation that occurred, say, in a muscle of the flipper. And as a result of that, he passes his gene on. It's a very simple story, a very, alluring story, and it's very believable on a certain level. But as you start to get into the molecular level, it's really not working out. In fact, most of the time when they show these mutations, they have to do this. And Stephen Gould questioned neo-Darwinism. He said, all you guys are doing is just waving your arms and giving us just so stories. a mutation will happen and then assume 1 billion years of things. Or like on the right-hand side here, the two forest rangers notice that the bear is carrying a crowbar and a drill because the garbage container caused it to get smarter, and natural selection gave it a higher IQ. That may seem post hoc and funny, but that's actually along the line of some of these papers I've seen. But then biologists had to spoil the ride. In the last 20 years, over 1,000 studies has proved that there's a codon bias, not just random codons, questioning Crick's frozen accident. These redundant codons we thought were just redundant are actually designed, and they have a code, a second code in the DNA. As we started studying codons amongst different things like the humans, the fruit fly, and E. coli, the bacteria, you can notice that the codon percentages are all different. Why is that the case? Shouldn't they just be random per neo-Darwinism? They discovered, however, that optimal codons, which are codons that don't have, that have the same synonymous setup without the synonymous mutations to different codons, they have the best codons, and they form a functional protein, as it shows in the top of the diagram. Whereas when you have non-optimal codons, it misfolds the protein. So even though you're making the same protein, it's not folding correctly, and therefore the fitness of the cell is actually off. And we were measuring all this time the proteins that were coming out, but we were not noticing the fact that they were not folding correctly. And so that's part of the lack of curiosity that we had on the frozen accident. This article came out just last summer. There's been a couple nuclear bomb articles that have demolished neo-Darwinism in the last year, and this is one of them. And it's called Synonymous Mutations in yeast are mostly strongly non-neutral. Basically, they're harmful. You recall this diagram, which I showed earlier, where you had synonymous mutations with different lettered codons. Say the leucine in the top left is a UUA, and then it goes to UUG. So both UUA and UUG can make leucine, but it's not necessarily an optimal codon. It's the same, it makes the same amino acid, but it doesn't fold the protein correctly. And that's what they were basically discovering, that the proteins were not folding correctly. And as a result, you want to have exactly the right codons. This means there's a second code in the DNA molecule. The authors claim that 60 years of evolutionary theory is challenged by declaring that most silent mutations are not neutral as previously thought, but rather harmful. That statement right there, it's like you could take that to a biochem class and say 60 years are out the window, guys. I mean, we thought that these silent mutations were neutral. And because they were neutral, you could calculate the level of natural selection using their equations and everything. Their equations are completely wrong. All of them, even phylogeny trees and other things are wrong because they were counting these mutations as to whether they were synonymous and non-synonymous. And it all goes back to Francis Crick. They say in their article, this requires a re-examination. I mean, that's a big statement of numerous biological conclusions about mutation selection. I mean, what? Mutation selection, that's the whole game. That we need to look at mutation selection, i.e. natural selection, effective population size. This is dealing with population genetics, which is where evolution comes out. Divergence times, that's the tree of life, and phylogenies, they're off. They're all off because of this. And disease mechanisms. We were ignoring disease mechanisms based on whether they were synonymous versus non-synonymous. That rely on the assumption that synonymous mutations are neutral. And that goes according to the central dogma. That paragraph there is huge. I mean, if we had just had that about 40, 50 years ago, it could have saved us a ton of research. So The other thing about the central dogma is it assumed information flowed from the DNA to the protein without any extra genetic information. This is where epigenetics comes in, and we'll discuss this next time, but I'll also talk about it a little bit in a second here. This idea is actually just now hitting the college. I mean, I came back from visiting with some friends over at a cigar place, and there was a nurse there, and she was looking at histones. And I said, "Oh wow, they're teaching about histones. She's going to be a nurse, nurse practitioner." And I said, "You don't know how important this stuff is. I mean, they're just now getting this." The problem is that
me. Dr. Mancha, can you mute your mic, please? Thank you.
Me?
No, other people.
Okay. Here's the problem. The problem is
somebody needs to mute Pamela's iPhone. Sorry.
That's better. The problem is Darwin's natural selection is irrevocably tied to the central dogma. I mean, it's irrevocably tied to it. Its whole basis is tied to it. And as Gandalf says, you shall not pass. You know, these older guys that are old neo-Darwinists that are still living around the university and chasing young ladies, you know, they don't want to believe the new stuff that's coming along. But what's encouraging is the younger people do believe it. There was and the Royal Academy had a meeting a couple of years ago, and one of the lead speakers who's pressing the need for to put epigenetics, you know, into the mix. And somebody stood up and said, "Isn't this like a culture war between the old neo-Darwinists and the new young guys that want epigenetics?" He said, "Yeah, it's a culture war, and one that we're going to win." He said half the audience cheered. I mean, that never would have happened when I was in school. Everyone believed neo-Darwinism. But now everyone is bailing on it, especially the young people, which is actually kind of encouraging. This was also one of the mainstays that ruined biologists for a decade. I mean, for a generation of biologists, and that was Richard Dawkins' The Selfish Gene. He wrote his book in 1976, and then two papers in Nature, which is the top journal, came out in '80, taking up for the selfish gene theme with Orgel and Crick. So Crick tied basically his central dogma to the selfish gene. And think of what that does. The central dogma says DNA goes to RNA goes to protein. If it doesn't make protein, it's not in the central dogma. Therefore, 98% of the DNA doesn't make protein. We've ignored 98% of the DNA, which is astounding because of their need to stay with neo-Darwinism. And I'm going to give a little YouTube on this junk DNA that they ignored. Oops.
In 2001, researchers of the Human Genome Project determined that only 2% of human DNA are genes, the sequences that directly code the proteins our cells use to carry out their functions. The remaining 98% of the genome was concluded to be a biological wasteland. But it turns out that this supposed junk DNA is crucial for controlling gene activity, often acting as switches to turn genes on and off. These switches determine where and when a gene turns on. When a regulatory molecule binds with its corresponding switch on the DNA, it will cause the switch to flip and the gene to be activated. With the regulatory molecule bound to the switch, the gene will be expressed in greater amounts. When the regulatory molecule detaches from its switch, gene expression will stop. Typically, the gene will be surrounded by many switches that allow the gene to be turned on and off under the control of different regulatory molecules.
Now, that was, of course, very oversimplified, but the project, the Human Genome Project, and I'll show a slide up next, was led by Francis Crick, who's a theistic evolutionist, and he wrote a book about God and the DNA, and he was very critical of creationists in general, because he said, well, we have the same junk DNA as other animals, and therefore, God's just trying to confuse you. Well, he really had to put his foot down his throat after that statement, because he based his whole project, which took 11 years and $7 billion, and everybody was waiting for the result. They thought they were going to cure cancer right afterwards, and it was the biggest nothing ever. I mean, and the whole reason was he only focused on the 2% of the coding DNA. That's the part that comes out of the central dogma, the DNA to RNA to protein. The rest of it does not code for protein directly, but it codes for other factors that control the coding portions. So he was at a meeting in Chicago, and they caught up with him, and he made this statement. He said, in terms of junk DNA, we don't use that term anymore. It was pretty much a case of hubris to imagine that we could dispense with any part of the genome. Now, isn't that an incredible statement? I mean, he finally admits that that was hubris to imagine that we could dispense. That statement, to imagine that we could dispense, is basically saying to ignore the complexity of God in the DNA and just think that it would be junk with any part of the genome, as though we knew enough, i.e., we knew enough like God, basically. We were humans, we were applying neo-Darwinism, and we ignored 98%. It was a total flop as an experiment. And enough to say it wasn't functional. Most of the genome that we used to think was there for spacer turns out to be doing stuff. So at least he got it straight. This is part of the junk DNA. It's called microRNA, and it doesn't code for protein, but what it does do is it goes in there and splices regular RNA to cause different proteins to be coded for. So there's a lot of other players that have come along that we did not know about because of the central dogma commitment. Another thing that came about, and this came about really in the late 70s, was the discovering of what's called prions. But we discovered a bad version of prions. You heard of mad cow disease or kuru, brain diseases when you eat the brains of your buddy or whatever. And they discovered that the proteins could be altered by the proteins of the disease, and it would change their shape and make it into an illness. So this was a protein to protein transfer of information, which violates the central dogma. Central dogma is DNA to RNA to protein, never protein to protein. Well, this violated, but they thought, well, this is just a one-off. It's not that big a deal. Later on, I'll show you how big a deal it is. They then realized that prions are also very similar to particular proteins, well, actually called intrinsically disordered proteins. And in fact, half our proteins in our bodies have intrinsic disordered proteins. And I'll show you a picture a little bit later. But these proteins can go in there and change the activation state of the chromatin, or the stuff that sits on our DNA, to activate it for hundreds of generations. So basically, you see here a protein to protein interaction, not a DNA to RNA to protein interaction, that's occurring for hundreds of generations, in total violation of the central dogma. You do recall that with neo-Darwinism, they counted on structured proteins too. And that's what I learned when I was in biochemistry. And now we know that, when I mentioned this earlier, that half our proteins are unstructured. They're called intrinsically disordered proteins, and they defy mutations in natural selection. And I'll show you why. Here we have a structured protein, which we used to think was the way things are, and half of our proteins are like this. They're very structured, and one amino acid off, and the whole thing can be messed up. Instead, we have 51%, slightly more than half, is made up of intrinsically disordered proteins, and they have regions called intrinsically disordered regions. So you can see the what looks like a phone cord in the middle protein, and that region is able to flop back and forth and move the active portions of the protein to interact with DNA and RNA and fats and all kinds of things. And they can also assume a prion configuration, which we saw before. So intrinsically disordered proteins, but here's the fascinating thing. Because they flop around like they do, if a mutation occurs in the area that flops around, it really doesn't affect its function. So basically, mutations don't change its overall function. Therefore, natural selection is not working on this. Natural selection is not working on 50% of our DNA. Intrinsically disordered proteins are conserved across different species over tens of millions of years. No evolution for millions of years. They literally can compare these things from bacteria to humans and the types of proteins and look for similar genes, and they've discovered that they can have the same function even though there may be mutations in there, because it doesn't matter whether it's mutations, because the things can flop around anyway. I mean, that's really death knell right there. That completely disproves neo-Darwinism. Here's a picture of one. You can see that the areas that bend are the intrinsically disordered regions, so that they allow for the bending to occur. And then you see the little different spots that are thin pencil-like areas, and they can interact with different molecules and cause different effects. Due to their flexibility, prions can act like structured proteins or intrinsically disordered proteins. I think we saw that earlier. You see on the far right, far left, it's more intrinsically disordered, and then on the far right, it can take a structured protein as well. This is a killer paragraph. Prions, or intrinsically disordered proteins, are a paradigm shifting mechanism of inheritance. Paradigm shifting. I mean, it's all changed, guys. Neo-Darwinism isn't working. This is a paradigm change in the mechanism of inheritance, in which the phenotypes, the outward shape of the body, is coded by self-templating protein conformations rather than nucleic acids. In other words, the proteins are able to change the outside without nucleic acids. And this is talking about intrinsically disordered proteins drive emergence and inheritance of biological traits. I mean, that's it. If it's causing the inheritance of traits and it falls outside of the central dogma, then it basically falls outside of neo-Darwinism. So what happens with the central dogma? It's caused several different things. Number one, it ignored the reason for the frozen accident in Crick's genetic code. He was uncurious to quickly accept a neo-Darwinian explanation. Number two, it wrongly concorded synonymous codons with neutral selection in a post hoc fashion to prove neo-Darwinism. That's those different types of mutations we talked about, the synonymous versus non-synonymous. Number three, it assumes that assuming the central dogma delayed discovery of the second code in the DNA. Isn't that incredible? The second code in the DNA, and that's the codon bias that we saw. Number four, the central dogma led us to ignore 98% of the DNA as junk DNA. I mean, if you were a physicist and your equation ignored 98% of what's going on out there, you'd be thinking, I want a better equation, or, you know, I want to try to get above 50%. But in order to ignore 98% of it, and then have Richard Dawkins, you know, one of the biggest atheists out there, write a book to try to explain it as being selfish anthropomorphically. You know, it's a selfish DNA. That's why it's hanging around. You know, I mean, that's not an explanation either, is it? I mean, the junk DNA was just selfish, and that's why it hung around. Number 5, ignore the possibility of prions or the intrinsically disordered proteins. Number 6, the central dogma delayed the discovery of epigenetics, and we'll talk about that later. Number 7, of course, is to be continued, and there are other many factors. So this is, you got your 2 simplistic views, the central dogma, DNA to RNA to protein. You can see it on the right as well. That leads to the genetic determinism that biologists have been declaring all this time. Or you have the central dogma with an overlay of epigenetics and all these other factors that you can see on there, much more complex picture. And with its computer programming, you have self-programming that's reprogramming itself as it's going along. And this is computer code that's actually reprogramming itself. So if you're interested in this sort of talk and some of these things, you can catch up to me on Darwin versus Lamarck. That's my Facebook page. And I publish articles about all these things that we've been discussing tonight. Of course, they're in much more detail. All right, that will conclude my talk.
Well, thank you so much, Dr. Mancha. There's a lot of stuff to be chewed on. Now we open up to any comment, questions.
Well, when you told me to try to just make it a biology 101, I said I tried to, and I just realized I can't. It's the central dogma is very complex, but I tried to keep it as simple as possible. But I'm glad you recorded it, because I think everyone might want to listen to it as a recording over to see what I talked about.
So had this material, had it gone down or trickled down to the college level biology?
Well, this one lady who was a nurse practitioner was looking at histones, which is involved with that. But the issue, for instance, in that Nature journal where it said synonymous mutations are harmful, which blows up 60 years of neo-Darwinism, they're really not hitting that probably unless you're in a graduate level. Biochemistry or molecular biology or something. They're still probably teaching the central dogma in biology 101, which is tragic, really. Really, it makes you make all these conclusions that are completely inaccurate. It seems so neat and easy,
So in the professional sciences, is Darwinism no longer settled science?
If you do a PubMed, if you search for neo-Darwinism, there was only 5 articles last year, and one of the articles completely lambasted it. It said, and it's in my Darwin versus Lamarck, but it basically says neo-Darwin needs to mutate to survive, and it basically says it's doing nothing. It went over the whole history, how it's not getting anywhere. In the last 10 years, there's 120,000 articles on epigenetics, and yet there's only about 200 articles on the modern synthesis, which is the theory of evolution. They really don't talk about it. They know it's dead, but they really don't talk about it.
Well, we know they're not going to run to creationism. What are their alternative models that they're presenting?
Well, that's the problem. What epigenetics and the things I talked to you tonight about are basically puts teleology back on the table. And they thought we were done with that with Darwin. We don't have teleology anymore. But suddenly you discover there's a second code in the DNA.
What is pediology?
Teleology, T-E-L-O-L-O-G-Y, design, basically.
Okay.
It's not intelligent design, but it is design.
It's working toward a goal, not just flopping around.
I'm sorry?
Teleology is working toward some predetermined goal.
Yeah, but it assumes that there's design features in that. But I would say that, and I've read several articles that are saying it's back on the table now because of these features. And I mean, just to think, DNA was so complex, and now we've got a second code sitting on top of the DNA, which we had no idea until about 20 years ago. These are codes upon codes that it makes it that much more irreducibly complex, you know, to use that term. You know, I just and then epigenetics sits on top of that, which is another code. The ribosome itself is almost a quantum computer in how it functions. They're discovering code upon code upon code, and it's getting to where this is just intolerable to say that it could happen on its own. Now, there are some people like Cy Garth, I don't know if you've seen him around, he's become a theistic evolutionist who is a very strong atheist, but has switched over. And the stuff they're talking about is this stuff is just too designed to think otherwise, you know. So I think that it's a great time. Man, I wish, although I'm glad I took my biology when I did, because it was so simple. You know, today is so complex, much more complex. But on the other hand, the stuff against neo-Darwinism is just amazing. When I was in college, neo-Darwinism was a fact like gravity. You couldn't question it. About the best I could do is I grabbed a hold of Moto Kimura's theory that he had, which I thought was better than Darwin. And then all this stuff started coming out about 10 years ago, and I started reading epigenetics and realizing this is the death nail. In other words, we don't need, for instance, we don't need these creationist groups to go out there and try to create arguments that are based on the old stuff. All they need to do is educate about epigenetics and the new stuff, and it does away with neo-Darwinism, which I think is simpler.
So your Facebook says Darwin versus Lamarck. What or who is Lamarck?
Jean-Baptiste Lamarck was 50 years before Darwin. And in some of his writings, the things that he describes was almost identical to what we see in epigenetics today. And a lot of people are calling epigenetics neo-Lamarckian. He influenced actually Darwin quite a bit. Darwin quoted him at least 20 times in his Origin of Species. In his second book, I forget the name of it, he basically said that Lamarck's mechanisms were more important than his. But the Darwinists all grabbed a hold of natural selection and survival of the fittest and went with that, and they specifically ran Lamarck off. In 1900, August Weismann, German embryologist who's brilliant, actually, he did this experiment where he cut the tails off of mice generation after generation. He said, well, you know, their tails are still there, therefore Lamarck doesn't work. But Lamarck didn't predict a mutilation experiment. He was saying, and the thing that Weismann missed is if he studied those mice, if you showed them a pair of clippers, they would go running because the behavior was carried on generation after generation. So the great grandchildren of that original mouse that got his tail cut off would go running at the sight of clippers, if you will. That's the part he missed, you know.
Dr. Mancha, see, Lamarck believed that environmentally acquired characteristics could be inherited in the next generation. Wasn't that his idea?
That's correct, and that's soft inheritance, we call that. And neo-Darwinists, August Weismann specifically went against that, as well as the modern synthesis.
Yes, I know. I've been indoctrinated in the modern synthesis myself.
Yeah. Well, and they said that this isn't happening. And even today, these hardcore Darwinists are arguing that that's not occurring, but there are so many experiments that are coming out that's disproving this, that they're finally kind of throwing their hands up and saying, okay. But I mean, I saw YouTube 6 months ago, this lady was saying, well, that's not really, you know, soft inheritance, you know, and they quip about it. And they think that epigenetics is a flash pan thing that's come along, and it's not. If it was flash pan, there wouldn't be 120,000 articles in 10 years. You know,
And
so that, you know, it's where it's at, especially cancer, you know.
in just a couple sentences, what is the science of epigenetics?
Well, we'll go over it next time, but epigenetics is basically, it comes from the Greek word or epi, above the gene. There's tags that sit above the DNA.
Okay.
To change how the DNA is expressed without changing the letters of the DNA.
Gotcha.
And we'll go over that next time in detail. The central dogma, by the way, ignored that possibility. It was just straight up DNA. There was nothing on top of it, you know. But epigenetics completely goes around the central dogma in a way. So that's another thing, and we'll talk. I'll mention the central dogma next time a little bit. But please do look over this thing, and if you have particular slides that you want me to, that you say, look, could you explain that a little better? I can use it to change my next talk.
Vaughn, you mentioned before that now, in addition to the genetic code, there are many other codes, including the epigenetic code.
Yeah.
I would be interested in knowing who has published a specific epigenetic code. Where is the epigenetic code?
Well, nobody's published it. It's so complex. They can't.
What makes you think then that the epigenetic code has all the answers to natural selection and neo-Darwinism?
Well, the reason is for the central dogma to work, you have to have DNA codes for RNA, codes for protein. But
yeah, and you've actually shown that does work.
What, the DNA, RNA, protein?
Yes.
Yeah, that does work for what, 2% of our DNA. The 98%, it doesn't work that way. So for 2%, yeah, the central dogma works for the majority. But on top of the DNA, now we have these tags that tells it not to make RNA. Or we have tags on the RNA that changes the way it's spliced and makes different proteins from it. Or we have epigenetic tags on proteins that's also changing things. So it's just a totally different game. And that's why it's, you know, it's apple and oranges to even try to compare this to the way it was. It's like we need to junk the whole idea. You know, when you say DNA, RNA, protein, it gives you this whole idea that it's just that simple, and it's not. It's really much more complex than that.
So do you think that neo-Darwinists today reject the central dogma?
Well, okay, there was an article I read, and I think I'll have it in the next talk, the Federalist, and they feel like at least 35% of the hardcore Darwinists now realize that neo-Darwinism has big problems.
So I'm sorry, 35% says it has problems, or says it doesn't have problems?
It does have problems.
Okay, so that implies that 65% say it doesn't have problems.
You're right. And these are older people.
that still is the predominant idea among biologists.
The thing is, though, you know, P.Z. Myers, who's a flame-throwing Darwinist, right? He bemoaned the fact that all his grad students are going into epigenetics. And that's because that's where the money for cancer research and other things is at. I mean, nobody gets a study to try to prove neo-Darwinism anymore, really. And it's leaking, you know, the ship is leaking badly as these new concepts come along. You know, we talked about the codon bias. We know there's a GC bias also in the DNA. That's another code. There's different repair mechanisms that cause things. I'm going to present an article out of Nature next time we meet that totally blows apart mutations and the way we thought about mutations. So all these things keep coming out, and that leads to that meeting I was telling you about, the Royal Society, and half the room broke out cheering for epigenetics, and I said that's probably the young people. You can go online and look at Houston or Harvard or whatever and look at their epigenetics program and look at the people, and they'll show them in all their white coats. and there isn't anybody over 30. I mean, they're all young and they're doing the work. They're pulling it. The older neo-Darwinists, yeah, they're teaching biology 101 or evolution or whatever, but they can't really show all this stuff anymore. They've got young whippersnappers that are coming in there and saying, what about, codon bias, or what about this? Doesn't that violate? He'll look like a total idiot. So he's going to have to change, try to absorb these, questions that are flying at him, so, but that, very hopeful actually.
Dr. Mancha, in that 2016 meeting in Cambridge that I think you're referring to, some of the insurgents, if I may use that word, were saying that one of the deficiencies of neo-Darwinism, it doesn't explain large-scale change in organisms. But I'm wondering if with epigenetics and with some of these other mechanisms, proteins affecting proteins directly and that sort of thing, that gives evolutionists even more ways in which things can change.
Yeah. Well, no, I mean, you would think so. I mean, they cast aside junk DNA only to wind up trying to say it's like it's their very own. And yet when you talk to them and say, wait, you were calling it junk, and they'll try to say, well, it's still mostly junk. And then you quote an article and they'll fight with you about that on how much junk is junk. And so their position, in my view, is pretty weak, actually. The research is going with the young people. It's going with these new fields. I would say intrinsic disordered protein research is where it's at. I mean, here are proteins that can withstand all the mutations you want to throw at it, and it still maintains its function over millions of years. That's a death knell to neo-Darwinism. Proteins, they need a structured protein for that to, a change to occur. But if you can absorb mutations, then that's antithetical to neo-Darwinism. So, I mean, basically what we see is a lot of little islands that they're starting to spread with hundreds of articles that are filling things in. And they're going to touch one another real soon. And we're going to start to see some really larger pictures. But I mean, I've been researching this stuff for 7 years ago, and the articles I was reading then, gave you a sense that you knew that everything was changing, but every year it just more and more was filling in. And I feel like this year, the stuff I read is just really starting to explain all that stuff. However, that being said, junk DNA is the 98% we ignored, and it's the stuff it's discovering it's doing is just unbelievable. And until we understand all that, it's for instance, our chromosomes are packed in the nucleosome in different zones, and that packing is completely dependent upon junk DNA. So how could it be that this stuff that was supposedly junk for millions of years is packing our chromosomes exactly correctly for life? There's just kind of questions like that need to be answered. But I think it's exciting. I would love to be in biology right now. I wouldn't be in biology though. I would be undergrad. I would be in biochemistry or molecular biology or something. But I wouldn't be straight biology.
So another thing I'm surprised that you haven't mentioned is in 2012, there was a follow-up study to the Human Genome Project called the ENCODE Project that indicated that their study showed that about 80% of the human genome is actually usable, not just 2%.
Yeah. Well, in fact, too, what they said is not only is that it's 80% that's functional, but that 80% is within 8 kilobytes of other functional elements. And that drives the percentage of usefulness up around 95%. And the Darwinists hate to hear that, and they fight over, how much of it is junk and it's not. And so, yeah, the ENCODE project, the ENCODE project, it had several thousand people that ran that thing in 400 labs. It took 11 years, and it has been cited. I just looked at it the other day, the paper. It's been cited by 11,000 articles. That's how, there's been no article that's been more impactful to this field of science than the ONCODE letter. 11,000 researchers cite them.
And yet, as you point out, 65% of biologists still cling to the neo-Darwinist theory.
Yeah, you're right. You're right. Well, I mean, it's like MacArthur, old generals never die, they just fade away. We're just gonna have to let these guys fade away. And,
well, I dare say that until someone comes up with the actual epigenetic code, you're not going to really pull away people who have clung to the neo-Darwinism idea for all their lives.
Well, the thing is what you're describing, and the way you put that question is the reason. Part of the answer is it's not a code like DNA with A, T, C's, and G's.
How do you know? You don't know that.
Well, no, I mean, what I mean by that is, for instance, epigenetics occurs in CPG islands more often than other areas, and then it attaches to histones and other things like that. But you're right, for somehow the body knows how and where to put these things, and it's influenced by the environment. There's so many moving parts and so many balls in the air that nobody so far figured that out, and I'm not sure if we ever will, but they're getting closer to knowing certain things about how it works. So, but DNA was simple compared to this stuff, really, if that makes sense.
Dr. Mancha, there's 2 questions on the chat here. They want to find out about, do you have a website? People cannot find it. And then
Well,
the second,
actually, I don't have a website. I have this Darwin versus Lamarck. And there's about 400 folks in it. And then I have some YouTubes.
And that's on Facebook?
Yeah. And then I have some YouTubes, which you can look at things. I'll probably put this lecture on YouTube. And I've got some other lectures I've given.
and the second chat question is from Ron Martin. I wonder if epigenetics are part of an uncoding DNA, or is it in material coding the DNA chains?
Well,
Sure.
okay, the junk DNA, which we talked about, that makes up 90% of things. The junk DNA is able to shift genes around like shuffling cards, and all that is done with epigenetics. And epigenetics promotes or demotes the junk DNA to let it move around. So it's like a computer that's reprogramming itself as it goes along, and it's all due to the epigenetic interaction with this non-coding DNA. So epigenetics is not only working on the coding DNA, but the non-coding as well. And what's more important is when they do different comparisons across the different scales of life, from bacteria to human beings, they've discovered that epigenetics goes all the way back to the very beginning of life. It was always there, in other words. It didn't just come along, so, and that's just blowing their minds. How in the world could this have been there? And believe it or not, biologists discovered one of the epigenetic chemicals, methylcycline, back in 1925, before the DNA molecule was discovered. And I mean, that's 100 years ago. The clue to epigenetics was around 100 years ago, and we ignored it basically because of our models of neo-Darwinism. So it's really quite tragic.
Okay, at this point, I'm going to stop the recording because it's all question and answer, but I want to continue the conversation.